Good evening. I want to thank Professor Farbman for inviting me to this program. And my job this evening, as I understand it, is to summarize some general information on functional imaging in a way that will provide some background material for Professor Farah's lecture. I hope that my comments this evening are not going to be too technical for those of you who've never heard of functional imaging, or too simplistic for those of you who have. And I realize, of course, it may not be possible to reach both goals at the same time. So let's get started and see how things go. The protagonist this evening is the human brain. And here, for example, we're looking at the human brain from the side, and just the covering of the human brain, which is known as the cerebral cortex, contains approximately 40 billion of these building blocks that you see here, which are known as neurons. And each neuron contains from about 1,000 to 10,000 connections through which it communicates with other neurons. So the total number of connections, also known as synapses, in just the surface of the human brain is in the order of 10 to the 14th power. It's 10 with 14 zeros. Very big number. It's a larger number than the total number of stars in our galaxy. So the brain could be conceptualized as a connectivity machine, as a networking device. And this is the organ that allows us to think, to feel, to run, to remember, and so on. How is the brain organized? How is this function made possible? And this is a question that's been addressed in many different ways. Almost 200 years ago, a very charismatic man known as Franz Joseph Gall suggested that bumps on the skull that he could feel with his bare fingers actually reflected the specializations of the immediately underlying area of the cerebral cortex for very complex functions such as secretiveness, acquisitiveness, cautiousness, sublimity, and so on. One person who took this very seriously was Henry Lavery of Wisconsin, who in 1905 patented this contraption known as the psychograph. The psychograph measured the bumps on the skull with 160 probes, and then on the basis of those measurements, then automatically printed out this beautiful chart where 32 mental faculties of that individual were rated on a scale from weak to very strong. In the 1934 Chicago World Fair, hundreds of people waited in line to buy tickets so they could have their heads examined by the psychograph. And in 1934, the psychograph netted $200,000, which in today's money must be a fortune. Wouldn't it be nice if we could all adopt this business model? I'm sure Vice President Moore would be very pleased to see the various functional imaging enterprises in the university turn from cost centers into sources of revenue. But it's also fair to say that this phrenological approach to brain function led to a dead end. And the real roots of our field can be traced to the careful clinical examination of patients with focal brain injury. And let me first tell you about the case of Phineas Gage, who sustained a horrible accident in 1848 in Cavendish, Vermont. And according to Dr. Harlow, who reported this case, before this accident, Phineas Gage had all the virtues that you would have expected from a New England Yankee. He was provident, prudent, thrifty, he loved his family, he was loyal to his company, he kept the Sabbath, and so on and so forth. But one day, Phineas Gage was unable to get out of the way of a dynamite explosion, and an iron tamping rod went right through his head. It entered under the cheekbone that you see here and exited from the top of the skull, in the process chopping off the front part of the brain, or the front lobes. Now, everybody thought Phineas would die, but under the watchful eye of Dr. Harlow, he actually recovered and left the hospital on his own 2 feet. And there was a lot of discussion among the Boston medical circles to the point where illustrious Harvard surgeon Dr. Bigelow publicly started to wonder whether the front lobes in the human brain served any useful function whatsoever. And around that time, John Harlow pointed out that while Phineas Gage looked physically healthy, that his personality had changed radically. Phineas Gage started to ignore his family. He would disappear from work and turn up days later in a drunken stupor. He took up gambling. Worst of all, he started to use profanity. He became inconsiderate. He had no remorse for hurting other people's feelings. He made bad decisions, and he wandered around the globe penniless and homeless until he landed in San Francisco, where he died during an epileptic fit. Gage's mother told Harlow about the death. Dr. Harlow went to San Francisco, and with the help of the mayor of San Francisco, had the body exhumed and the skull sent to Harvard Medical School for safekeeping, where you can still see the skull where this picture was taken. So the case of Phineas Gage, then, has taught us that the front lobes of the human brain are critical for the most refined aspects of mental function, such as moral judgment, conscience, insight, foresight, stick-to-itiveness, inhibiting inappropriate behaviors, and so on and so forth. Now, the second case I want to tell you about occurred in Paris in 1861, when one of Paul Broca's patients had a stroke, a cerebrovascular accident that destroyed this part of the brain right here, which is still known as Broca's area, which is right behind the part of the brain destroyed in Phineas Gage's brain. Paul Broca's patient did not show any change of personality as a result of this stroke, but instead he lost the ability to express himself in spoken or written language. Broca then looked at many other patients with brain lesions, and reluctantly he had to conclude that language was not a generalized function of the brain, but there was one part of the brain located right here in the left side, but not the right side, that was more critical for language than any other part of the brain. So this was really a revolutionary thought. The fact that language could not only be localized, but that it was also asymmetrically localized to the left side of the brain. The third case I want to tell you about takes us back to New England, Connecticut. A young man called H.M. has a bicycle accident, hits his head, and then develops intractable epilepsy. Dr. Scoville, who is a surgeon, traces the source of the epileptic discharges in the brain to a small region here, deep in the brain, known as the hippocampus. And then, since this epilepsy can't be treated medically, Scoville decides to surgically remove the hippocampus. Many years later, H.M. had an MR scan of his brain, and here you see where the normal person has hippocampus, there's just a big black hole. So the surgeon was very successful in doing what he had wanted to do. And in fact, the surgery helped to treat the epileptic fits. But Brenda Milner, who was the neuropsychologist working with Scoville, noted that while the epileptic fits had decreased, that H.M. had also lost his capacity for memory. He was unable to remember what happened a few minutes ago, a few days ago, and he could not learn any new experience. So here is another revolutionary finding. Something as pervasive as memory is critically dependent on a little piece of brain tucked deep inside the brain right here called the hippocampus. Now, these three cases that I told you about have been milestones in our understanding of brain organization. But I think you will agree with me that a field where the senior scientists have to do nothing but idly wait for something bad to happen to their patients' brains so they can report it in the scientific journal was not destined for very rapid progress. And in fact, people started to wonder whether we could ever learn anything useful about brain organization in normal people, people who had no brain injury. And this takes me then to functional imaging. For many years, physiologists had known that physical exertion made the heart pump more blood to the muscles in order to replenish the nutrients that the muscles were using up. So the question came up whether the same thing was true in the brain. Did increased mental activity require more blood? This is a question that was asked by Eugène Gley, who was a professor of physiology in the University of Nancy in France, and he put a little diaphragm on the neck on top of the carotid artery, which connects the heart to the brain, and then measured the amplitude of the pulse on a kymographic drum. And Gley found out that the amplitude of the pulse, which is proportional to the amount of blood that goes from the heart to the brain, was greater when his subjects were engaged in some mental activity, mental exertion, than when they were at rest. And then, tongue-in-cheek, Gley told his colleagues in natural philosophy, as they were called then, that the increase in the carotid pulse was not as much when the subjects were solving mathematical problems than when he had asked them to ponder on philosophical dilemmas. Now, the carotid artery is the main pipeline that sends blood from the heart to the brain. But in the brain, the blood is then distributed further to the tissue by this amazing net of fine vessels called capillaries and arterioles. So there really is no general sprinkler system in the brain. Instead, almost each blade of grass, each at least tuft of grass, has a specialized private delivery system. Let's assume that you engaged in some mental activity that requires the activation of neurons or the participation of neurons in this area right here. This additional work of the neurons then triggers the need for additional oxygen and glucose that needs to be delivered by increasing the blood flow just to that area of the brain. The increase in blood flow is only in the order of 1 to 2%. But the influx of this additional oxygenated blood changes the magnetic properties of the tissue in this region. And through some almost miraculous developments in magnetic resonance imaging, we can now detect this local change in blood flow from outside the skull and display it on a three-dimensional rendering of that individual's brain, as you can see in the case of this person who was just moving her fingers up and down. And you see this beautiful area of activation right on top of the motor and sensory areas controlling the fingers in her brain. And even though she looks painfully scalped, I can assure you that this scalping is just computational, it's virtual, and she is currently enjoying her brain just as much as she did before this experiment. Functional imaging is really quite straightforward. The subject lies on his or her back, then the head is eased into the scanning device, into the coil, and we then present the subject with a task we want the person to do, either through a mirror, which is located right here, or through earphones. And then we give the subject a response box so that they can push buttons to tell us their response as they solve the task. And really, this is quite a remarkable method. This is a method that allows us to look right through the hair, through the scalp, through the skull, look at the surface of a brain as if we were holding it in our hands, and not only that, but also show us just those parts of the brain that are activated by the task that we have given the subject. And all of this with a temporal resolution of about a second and a spatial resolution of about a millimeter. Let's take a look at this experiment, for example, where the subject is asked to read a word that is presented in the left visual field. Now, as you may or may not know, visual pathways are crossed in the brain. So this very hot area of activation that you see right here is right on top of visual areas in the right side of the brain which receive this visual information. Now let's see what the brain does with this information. Now, as you look at the right side of the brain, you see that things are pretty silent. So the right side of the brain is not all that interested in reading. Now, when you go to the left side of the brain, you see these fireworks. There is this activation all over the place, but especially over Broca's area. So here then, in one subject, we confirm Broca's idea that language is localized and asymmetrically localized to the left side of the brain. Now, this much we could have learned by examining patients with brain injury. But functional imaging can give us insights into brain organization that even thousands of years of observations on patients with focal injury could not have yielded. And let me give you one example based on the work of Dehaene and colleagues in Paris. This was a work done on reading. And it turns out that reading could be conceptualized as a three-step process. The first step occurs right here in these blue areas. These are very close to the visual areas I just showed you in the previous slide. And nerve cells in these blue areas give different responses to the same word if it is written in a different font. So these are the kinds of neurons that encode the word at its most superficial level as a shape. The second stage occurs in this orange area, which is just in front of these blue areas. And here we have neurons that give identical responses to these two words, even though they are written in different fonts. So these neurons then are able to do a deeper level of encoding where a word is encoded as a word, not just as shape. The third stage occurs when the subject is asked to process the meaning of the word, and here you have this explosion of areas that show activity. Now, this may initially appear to be chaotic, but there is a method to the madness, and let's see if we can reconstruct the process. So step number one is for the visual information to be conveyed to the part of the brain where words are encoded as words. Once the visual event is identified as a word, then this information holds the key to open the door to Broca's area, which is really the major gateway into language processing. From Broca's area, the information is conveyed to these multiple regions which collectively encode the meaning of the word butterfly. So what we have here is not a comprehension or meaning center or a butterfly center, instead a distributed network which collectively leads the march from word as shape to word as meaning. Now, this network approach also applies to other mental functions, such as memory. This is an experiment that was done here at the Northwestern Cognitive Brain Mapping Group by Paul Reber and Ken Paller. And while these subjects were being scanned, they were given two word lists. One list they were told to exert an effort to memorize, the other list they were told to forget. Now, what the subjects didn't know is that after scanning, their memories for these words were tested for both lists, both for the list where they were told to memorize and the list they were told to forget. And there were two areas where neurons became active during this task. In the front lobe, the same region that was damaged in Phineas Gage, and in the hippocampus, the same area that was surgically removed in HM. But the pattern of neural responses were quite different. In the frontal lobe, the neurons gave a much greater response in response to those words that they were told to effortfully remember, to memorize, than to the words they were told to forget. The hippocampus did not show such a distinction. Instead, it turns out that the hippocampus gave a much stronger response to those words that later turned out to be remembered successfully, whether or not they were in the remember or forget list. So what you have here then is a collaboration of these two areas. The frontal lobe, which is important in mediating the effort, concentration, attention that you need during voluntary memorizing, and then the hippocampus, which is very important for making sure that the filing system is in good order. And in clinical practice, we see memory disturbances in patients that have either frontal lobe damage or hippocampal damage, but from different reasons, as you can see here. One of the most important things that we do in everyday life is to identify the value of events and experiences we encounter, so that we can pay attention to things that are important and ignore those that are not. And this, too, is a part of brain function. In this experiment, which was also done here, we asked our subjects to come to the scanning session after an 8-hour fast, when they were really hungry. And then we showed them in the scanner pictures of foods or pictures of tools. Then we took our subjects out of the scanner and we fed them a sumptuous feast of their preference. Whatever they wanted, they had. Then we put them back in the scanner and again showed them pictures of foods and pictures of tools. And we found that there was one little area right here in the region called the amygdala, where nerve cells gave a much more enthusiastic response to pictures of foods when hungry than when full. And no such distinction was made by the amygdala to tools. So here then is a piece of the brain where neurons are capable of encoding real-time shifts in the motivational value of a visual experience. And it's clear that this kind of brain area is crucial for directing attention, emotion, motivation to events and experiences that are important. Another part of the brain that plays an important role in encoding value is a region known as orbitofrontal cortex. And from many different experiments, we knew that neurons in the inner part of orbitofrontal cortex, a region known as medial orbitofrontal cortex, became more active in response to desirable, pleasurable events, whereas neurons in the outer part of orbitofrontal cortex, also known as lateral orbitofrontal cortex, became more active when the event was either repulsive or aversive. In this experiment, Dana Small gave her subjects a little piece of chocolate that she put on their tongues. And in response to the first bite of chocolate, this was the pattern of activation. It was in medial orbitofrontal cortex, denoting this as a very desirable event. But Dana didn't stop there. She kept feeding her subjects chocolate until just the thought of chocolate was disgusting, until they could no longer bear the thought or taste of chocolate. And see what happens. At that point, the taste of chocolate causes activation in exactly the other part of orbitofrontal cortex, in the lateral part of orbitofrontal cortex, which then denotes this as an aversive experience. Now, I find this really quite astounding. Here is exactly the same physical reality, a piece of chocolate on your tongue. And yet, it gives rise to very different patterns of neural responding, depending on the significance of that event to the individual. This encoding of pain and pleasure in orbitofrontal cortex is not confined to foods. In this experiment, subjects were given all kinds of faces and asked them to rate as attractive or not attractive. And it turned out that neurons that responded to faces that were rated as attractive were located in medial orbitofrontal cortex, exactly on top of the part of the brain that gave a response to the first bite of chocolate when it was pleasant. But there are also glitches in this system. In this experiment, the new mothers were put in a scanner, and they were shown pictures of their babies and pictures of other people's babies. And the experimenters wanted to know if there was a part in the brain that gave a more enthusiastic response to your own baby than to other people's babies. And guess what happened? Unfortunately, the part that gave a better response to your own baby was in lateral orbitofrontal cortex, in the part of the brain that also gave a response when chocolate was disgusting. Now, this is not necessarily a bad experiment. It just simply tells us that we should be careful, give the brain some credit for subtlety, and not try to really simplify either experiments or their interpretations. One of my favorite experiments in this group is this one, which was published in the first Valentine's Day of the new millennium. Now here the experimenters put people in a magnet and showed them pictures of families, family members or friends, and also pictures of the person that they were currently in love with. And they found out that these patches in the brain contained nerve cells that gave a much more vigorous response when looking at the person that you are currently in love with. So who said that functional imaging couldn't have practical uses? Now, if you kind of are in doubt about whether somebody is in love with you, you can stop plucking daisies. Now, all you have to do is to take a few pictures of yourself and your friend to the neighborhood functional imager, and you're going to have an immediate and precise biological answer. Now, the encoding of value could also have some commercial implications. This experiment had two phases. In the first phase, the subjects either saw a can of Pepsi or a flashing light, each of which signaled the delivery of a little drop of soda in your mouth. What the subjects didn't know is that in both instances, the soda that was delivered came from the same bottle of Pepsi. Now, in the second phase, the subjects either saw a can of Coke or a flashing light, and again, these signaled the delivery of a drop of soda, and in both instances, the soda came from the same bottle of Coke. And the experimenters asked the following question: does the brain react differently to a drop of soda preceded by looking at a can than to a drop of soda preceded just by the flashing light? And as common sense would dictate, in the case of Pepsi, the answer was no, that the brain was not fooled, that it detected that both drops of soda were the same taste and gave exactly identical responses. But not so with Coke. It turned out that there are all these patches here indicating that the brain actually interpreted the drop of Coke preceded by a view of a can of Coke as more pleasurable than the same drop of soda preceded by a flashing light. So here then is proof positive that this particular logo is adding value to the product. So advertising agencies now, I guess, could stop all kinds of questionnaires and canvassing and sales reviews, and all they have to do is buy a scanner for $3 million, and they would have scientific proof that what they are doing is of value. Now, functional imaging can also address issues of individual differences in preferences, talents, and so on. Now, as you know, animals, insects are supposed to have something called a pheromone that makes an insect irresistible to an insect of the opposite sex. It's been debated whether humans have pheromones, but some people believe that there is a substance in male sweat and in female urine which plays the role of pheromones. So Dr. Savage and colleagues actually scanned men and women as they were sniffing male sweat and female urine. I hope they were paid well. The results, but were fascinating. it turned out that there's a region here called the hypothalamus, which is really a key part of the brain dealing with visceral and hormonal control, which in women's brain was much more active while sniffing male sweat than while sniffing female urine. And in men, it's just the opposite. In men, the hypothalamus is silent when sniffing male sweat, very active when smelling female urine. Now, when they look at homosexual men, the pattern was different. Here, there was a vigorous response in the hypothalamus in response to male sweat, and still a response, but less so in sniffing female urine. Now, the question, of course, is whether these men actually have this particular sexual preference because their hypothalamus is constructed in this way, or whether the hypothalamus gives this response because of their sexual experiences. And this is a sort of thing that functional imaging is in no position to address. Here we're looking at the brain of a gentleman called Rüdiger Gamm. Now, Rüdiger Gamm is able to calculate ninth powers and fifth roots with great accuracy, mentally, of course. And he can find the quotient of 2 primes to 60 decimal places. I don't even know what that means. I have never been able to balance my checkbook, and as far as I'm concerned, Rüdiger Gamm could be an alien from Mars, but he's obviously very good with mental calculations. So they put Rüdiger Gamm in a scanner and gave him just simple calculations. These green areas are the parts of Rüdiger Gamm's brain that were activated while he was calculating, but these are also the areas that are also activated in every other mortal performing calculations. But then there are these red areas that were activated only in Rüdiger Gamm's brain when performing calculations. So in some way or another, he is able to mobilize more of his brain while doing the same kind of calculations that other people are doing. So this, of course, brings up the possibility that perhaps functional imaging could at some point provide some sort of an aptitude test, perhaps even matching people with the appropriate careers. And of course, a related question comes up of whether there are things that a career or a profession does to your brain. And this is a question that was addressed by Eleanor Maguire. Some years ago, Eleanor Maguire, for reasons that are not clear to me, decided to do an experiment only on London cabbies. So she collected London cabbies, put them in a scanner, and asked them to mentally reconstruct a route of how they would take a customer who, say, got into their cab in Piccadilly Square, wanted to go to Victoria Station, for example. So this obviously required spatial memory, and she found in the scanner that while the cabbies were doing that, the hippocampus was very active, as you would have predicted from what I told you. So her next experiment was then to take 2 groups of people. She took cabbies, and then she took another group of people who were same age, same education, same background, but not cabbies. And she found out that, lo and behold, the cabbies had a larger hippocampus. Now, since it's quite unlikely that only people who are born with big hippocampuses apply to become cabbies, the implication here is that working as a cabbie, which taxes your memory function, caused a bigger increase in size, hypertrophy of the hippocampus, which is the part of the brain that is very important for memory. This is like building your muscles by pumping iron. So, and who knows, perhaps this is one reason why workaholics, for example, get such frequent headaches. But as I was looking at this, it just occurs to me that wouldn't it be nice if we made imaging available to all entering freshmen, and also after they graduated, so that long-suffering parents could have objective evidence that the tuition they are paying is adding real value in their progeny's brains that can be measured by ounces, centimeters, and so on. So that's maybe a future use of functional imaging. Now, then there are some other functional imaging experiments that I find particularly difficult to interpret, I must say. In this experiment, subjects were asked to make either utilitarian or non-utilitarian moral judgments. And the experimenters found this region right here in the right frontal lobe that was more active while performing utilitarian judgments. Now, I have no reason to criticize this experiment, but I find it a bit implausible that all the incredible complexities of moral relativism would be confined to a few flecks of brain tissue right here. I just somehow don't think that we are going to discover a John Stuart Mill center right here in the brain. This is another experiment I find interesting. Now this is really remarkable. It just goes to show you what lengths investigators will go. Here, the investigator stalked his subjects all day long and made notes about what the subject did. Then the subject was brought to the scanner and was quizzed on his activities of that day. But if a question was followed by a green light, the subject was instructed to respond truthfully, and if the question was followed by a red light, the subject was instructed to lie. And it turned out that these little red and yellow areas here were more active during lying. Now again, I'm a little disappointed at this result, since I thought that it really would have taken a much more brain substance to lie than just these little dots that you see here. On the other hand, this is the sort of news that the media really likes, and this paper was followed by this bold news that brain imaging can spot liars. And this is one area where I think it's our job as scientists in this field to really educate the public that these kinds of jumps are just simply not justified, and at least in my opinion, functional imaging is nowhere near becoming a lie detector, at least in its current state. And let me tell you, as my final example, this experiment. Now here, subjects were put in a scanner, and all they did was watch this professional actor lift this box again and again and again. However, in some events, the actor actually was lifting a truly heavy box. In other events, the box was empty, and the actor faked that he or she was lifting a heavy box, and the subjects were asked to detect the faked versus the true heavy lifting. And the result was these little yellow areas here, where nerve cells were more active when the subject was successfully detecting deceit. I must admit I'm totally baffled by what the purpose of this experiment is, or what it tells us about brain function, unless, of course, one is trying to construct an entrance examination for people who apply for the FBI or CIA or some such thing. So that's really all the time that I have this evening, and I hope I've been able to convey to you really my enthusiasm for functional imaging. This is a revolution. The field has changed. It's never going to be the same. But as in all other revolutionary movements, there are going to be some examples of excessive zeal. So there's going to be the alpha pluses and gamma minuses of functional imaging. And as the field matures, we're going to have to learn that not all questions can be addressed by functional imaging, and also that not all answers are interpretable. In the meantime, however, I think we're in for a most incredible ride into uncharted territory about how the human brain works and how it is related to the human mind. Thank you.
When you think about what the frontal lobe does, that is kind of, it's mission control almost for the brain. It's the seat of the soul, as Descartes envisioned, well, at the pineal gland, but it's like that. How good is the functional imaging method in determining whether a person will become a sociopath depending on the structure, the anatomy of his frontal lobe? I mean, is it true that all sociopaths have a deficient frontal lobe, for example, or a damaged frontal lobe? Can you make any predictions?
I really must say that I don't know what the answer is unless I try to define terms that there are so many different ways to become a good sociopath. In fact, in many instances, to be a good sociopath, you have to have a very, very good brain. There are some other kinds of sociopaths where things are just simply impulsive. For example, Phineas Gage was one of those sociopaths where he did things without thinking, without really considering the consequences. He didn't plot to rob banks and so on. There, the sociopathy is due to brain damage. But in some others, I would think that probably we should look for people whose front lobes are hypertrophied and who can really become good sociopaths.
So what can we say about Osama bin Laden's frontal lobes?
You know, they may really look indistinguishable from anybody else in this room.
Too bad. Yes, here.
How do you neutralize the distraction caused by periodic bang, bang, bang in the MRI?
You don't. Yes, the question is how, those of you who are not familiar with functional MR, how many people here had MR scans of one way or another? Yeah, quite a bit. The sound is deafening. And how do you deal with it? Two ways. First, we have earphones that absorb a great deal of the sound. But you still have this vibration that you can't screen out. But our subjects, essentially they get used, they adapt to it. We spend enough time in the magnet. The other thing is that in functional imaging, everything you see here is based on a subtraction, on a comparison of A to B. So in A, if they do a certain task, in B not, the sound is the same in both, so it tends to be subtracted from each other. And then there is an even more remarkable technological advance where you can actually write MR sequences so that things are quiet while you are doing the important task. And this is new development that is being used for certain kinds of experiments. So the answer is, the question is a good one and real one, but by and large the field feels that this is not a major factor in the results that we get.
Question back there?
Mainly used now instead of some of the other imaging techniques like PET or SPECT?
The question is whether functional MR is used diagnostically. Is that what you mean? Instead of, yeah. As far as I know, there is very little, if any, clinical use for functional MRI. This is still a very experimental procedure. There are a few exceptions, but by and large, this is not a clinical tool. In contrast, SPECT and PET have quite a number of clinical applications for diagnostic purposes.
Question in the back? Yes.
I have a question about the parsimony of the brain, if there is such a thing. You showed a few studies that dealt with the perception of reward or potential reward. In one study, there was amygdala activation when people saw hot dogs and they were hungry. In the other study, you had orbital cortex activation, either medial or lateral, depending on the design of the study. Other people find nucleus accumbens activation when people perceive monetary rewards, for instance. Is there hope that at some point we'll find that the same area sort of responds to all types of rewards, or are we going to find that depending on the task, depending on the stimulus, area X is active, and it's not the same area X across cues, across domains? And if we reach that state where basically the brain seems to be extremely specialized, for each cue there's one area that lights up, what do we learn? What do we gain?
I'm sorry, the last question was what?
If there is no parsimony in the way the brain deals with a large variety of cues, then what do we gain? If we find out that there are 1,000 little modules in the brain that deal with 1,000 different tasks.
Yes, that is the challenge, isn't it? that the brain is definitely not simple, and there is parsimony, but parsimony at a level that we will understand only when we can establish the rules. And right now, what is very clear is that there is no such thing as one location, one function. That's something that we left behind after phrenology. Right now, what we have in mind is that there are distributed networks for language, for perception, for memory, and many areas collaborate with each other. No area is devoted just to one thing. Someday it is, or in some functions, it's the key region for a certain function, and in the next function it becomes a participating area for some other purpose. And these are the rules that we are trying to establish right now. That's the way the brain is organized. We're not going to simplify it beyond this, because that's the reality. And this is why there are so many different methods we need to use. Functional imaging is just one. It's not going to be giving us all the answers.
Are there other questions? Yes. Dewey?
Over here.
Oh, here.
Doesn't Paul seem to fly in the face of the mind-body split, which has been important in the humanities in the West for centuries? How long do you think it's going to have some trickle-down effect to conventional wisdom, and can you say a little bit about that?
Trickle-down effect on what? I'm sorry.
On conventional wisdom, or on campuses, where people will or will not accept that.
It's like every area of science. I'm really, I think that there are facts and truths where the value is something that is based on the consumer. In other words, there's really no fight here about mind versus brain. But this is the way the brain works. Whether one wants to call it mind or not, that's an issue of definition. It's really like saying that any Picasso is made up only of pigment. Well, that's true, but not everybody who has pigment can make a Picasso. So there are reductionisms that lead nowhere, and these are the facts. What you make with them really depends on your background, orientation. That's really all I can say about that.
Yes.
I know that you don't believe very much about the correlation with people who are not telling the truth and brain imaging parts of the brain. But what substantive data have the proponents of this technique? What have they done to support this particular observation?
You see, what I'm really trying to say about the use of functional imaging in detection of lying is that the question is so complicated and this method is so cumbersome that this is not the right thing to do. This would be like, I can't even find an analogy, but it's not that it's wrong or right. It's just that this would be a very expensive, cumbersome, inefficient way to find out if somebody's lying. That's really all I'm saying.
Hi.
With a microphone.
It's a nice quiet place over here. In terms of selecting residents for orthopedic surgery, we can get an idea of the cognitive domain, somewhat the affective domain. Motor skills we don't have a good handle on at all. Do you have a test where you can scan someone's brain and tell whether they'd be a good surgeon, or should they go into one of the medical fields like neurology? Sorry, I had to throw that in.
Well, if we see any brain at all, we should steer them away from neurology towards orthopedics. I hope the tone of voice I used as I described that experiment indicated that I don't think that this is a legitimate question, simply because you can use your same brain to do different things, and there is, I am sad to say, no orthopedic center in the brain that you couldn't also use, say, to be a good sculptor or maybe lift weights.
Thank you.
Final question here. Yes.
I was wondering if there was any pattern in Alzheimer's patients that you see generally when you're doing mental imaging, brain imaging.
Yes, there are quite a number of patterns. One of them is the hippocampus, which is the memory area, is both smaller than in patients without Alzheimer's disease or people who don't have Alzheimer's disease at all. And also there are some patterns in the frontal lobe, in another region of the brain called the parietal lobe, which show less activity in Alzheimer's disease than in others. And these are quite striking differences. Today I didn't say anything about disease. I was talking about functioning of the normal brain.
So can you use diagnostically?
Yes, yes. Not functional imaging. You can use other scans, PET scan, SPECT scan, just structural MR. Functional imaging is really not a diagnostic tool at this time.
Thank you very much.